Welcome to this webinar. So my name is Julia, and I'm part of Cell Guidance System, which is the company who is sponsoring the webinar today. So just as a 1 minute quick introduction, I just wanted to introduce you our company. So we are a company based in Cambridge in the UK, and we also have offices in the US. More specifically in the Missouri, and we've been established since 2010. And since then, we have a lot of products and services in general, but for cell culture. So we have, for example, for plate coating, we have matrix protein, but we are also the distributor of Matrogen, which provide polyacrylamide hydrogels. We also offer growth factors, so in 2 different formats. We have a new technology, which is a sustained release over time, but we also have the recombinant standard one that you know already. We have different type of cell culture media as well, depending on your needs. So we have a photostable media. We also have another one for proliferation and maintenance of HPS seals. And you can also do custom media production. So you can give us your recipe, and then we can make the media for you. We also have a big part of services, including cytogenetics. So we are doing karyotyping services as well. And we have a large portfolio for exosome-related products. So we have for purification, profiling, detection, and tracking. So I just wanted to let you know that if you are interested in any of these products, you can always contact us. So through our website at www.cellgels.com. And I also added my email address. So it's julia.miliex@cellgels.com. But now we are really excited because today we are with Dr. Kevin Chalut from the University of Cambridge, who is going to present you today a new and the latest paper that has been published on Nature. And he's going to talk about mechanical signaling in stem cell aging. So if you have any questions during the presentation, you can ask them on the chat, and Kevin will answer later, because he will go on stage afterwards, and you can ask all the questions that you might have.
Thank you, Julia, and thank you to everyone for attending my webinar. I wanted to tell you about some recent work we did on mechanical biology of aging in the central nervous system and cell fate choice in general. In other words, I want to talk to you a little bit about why it's important, I think, when we're talking about stem cells, to think about mechanics. Starting with cell fate choice, and in fact with development, and development relies on a sort of coordinated cell fate choice. Getting the right cells to the right place at the right time. This happens all the time in development, and it tends to happen correctly. And even when you have an adult, you have to maintain tissue. And tissue maintenance also relies on cell fate choice to some extent, based on stem cells. Stem cells are needed to replenish the tissue appropriately for proper homeostasis. So all throughout the lifespan of the organism, you need cell fate choice to work correctly. And at the heart of cell fate choice is stem cells, our stem cells. And stem cells sort of have one of 2 things that they can do. It's quite simple in some sense. They can either self-renew, in other words, give rise to a copy of themselves, or they can differentiate and give rise to sort of differentiated tissue cells or more specialized cells. So they're a cell with exactly one choice to make. So in some sense, it's really quite simple. But underlying that sort of simplicity, or belying that simplicity, is the fact that even though all they can do is sort of self-renew or differentiate, somehow or another, they give rise to an entire organism, and they maintain the tissues within that organism. So again, cell fate choice, getting the right cell to the right place at the right time. And so my lab is working extensively on that. And, we're working on what underlies cell fate choice, which is a really complex interplay between signaling time and cell history. And we're trying to understand this interplay using physical biology, which can mean many things to many people, but to us it can mean one of two things, or the two things that we're interested in are the dynamics of cell fate choice. In other words, how do you get from A to B in a stem cell? But what I want to focus on today is mechanics. We firmly believe that mechanics and forces are an essential determinant in cell fate choice, whether or not it self-renews or differentiates, etc. So cells respond to forces. They are often attached to other cells. They're often embedded into extracellular matrix, or ECM for short. And within this sort of environment, they can spread. They can attach to other cells. They can migrate. There's many forces that exist within the environment. In fact, cells in general adapt, shape, and respond to their mechanical microenvironment. And we believe that's important for the following reason. So, many people will tell you, okay, well, biochemical signaling drives cell fate choice. And that is, of course, true to a great extent. And some people might even tell you the mechanical signaling drives cell fate choice, in other words, the mechanics of the microenvironment. But what we firmly believe is that it's the feedback between biochemical signaling and mechanical signaling that ultimately drives cell fate choice. In other words, if you give a cell or stem cell, for example, the exact same growth factors, stem cells are going to respond to those signals very differently in a stiff versus soft microenvironment. So how can we study mechanical signaling, especially in the context of other kinds of signaling? So one option is polyacrylamide hydrogels, or any sort of hydrogel in which you can control sort of the microenvironment. Now, one of the beautiful things about polyacrylamide hydrogels is that they have very tunable mechanical properties. You can tune them all the way from sort of embryo or brain stiffness, which is about 300 Pascal, to all the way to bone stiffness, which is very stiff, about 1 gigapascals, or a huge dynamic range within physiology. Now, for the hydrogels that we need, as I said in the title, we're working on central nervous system. This is a very soft tissue. And one of the things that we need to worry about is whether the polyacrylamide hydrogels give us good attachment of stem cells to the substrate, and we also need to be able to control for the extracellular matrix tethering and density. And so we have bespoke hydrogels that we use, which we call StemBond hydrogels. These actually use a sort of novel functional group that we bake into the hydrogel that allows us to completely control for the ECM tethering and density, and still be able to go all the way from brain stiffness all the way to bone stiffness. And we've used these hydrogels to culture stem cells from all sorts of soft tissue and do controlled studies of mechanical signaling. There are other hydrogels out there. This is just one that we used and worked well for the sorts of systems that we were doing. But again, it allows us to do very controlled studies of what happens to stem cells when you put them in a soft microenvironment versus a stiff microenvironment. And that was what was important to us here. So we've used them for significantly improved stem cell culture in embryonic lineages, liver, cartilage, etc. And we're starting to understand quite a bit about mechanics and normal stem cell function using these kinds of hydrogel systems, and how important mechanics is to cell fate choice. But what I want to talk to you about a little bit today is that what happens with mechanical signaling in stem cells when fate choice goes wrong. In other words, when stem cells cease to self-renew well, and when they cease to differentiate well. And one of the most notorious ways in which they start doing this is in aging. This is a sort of normal course of aging. The aged stem cells tend not to perform their simple choice well. They don't self-renew well. They don't differentiate well. And there's been many different theories as to why that might happen. As you can see on the right over here, you can have sort of excess DNA damage, loss of proteostasis, mitochondrial dysfunction. There's many sorts of cell intrinsic mechanisms that have been proposed that drive this sort of aging phenotype, this loss of function. So we were interested in oligodendrocytes. Oligodendrocytes myelinate axons. Now, myelin is essential for proper signal transduction in axons. It's essential for motor skills, cognition, etc. And if you lose it without replacing it, it gives rise to many pathologies. The most most well known as multiple sclerosis, for example, which is a demyelination disorder, which is exacerbated with age for the following reason. And that is that oligodendrocytes are supported by a stem cell or progenitor cell called oligodendrocyte progenitor cells, which I'll call OPCs for short. Now, OPCs are in the CNS. They're very numerous throughout the lifespan, and they give rise to oligodendrocytes. They're actually multipotent. They can give rise to other cells, but normally they give rise to oligodendrocytes, which go on to myelinate new or demyelinated axons. But like all other adult stem cells that we know of, they cease to function well as we age. And so this is an issue. So this sort of lack of regenerative potential, i.e. the inability to proliferate, self-renew, and differentiate, is a normal part of aging. And in this case, at least with OPCs, we hypothesize that it's driven by the microenvironment. And so this is all part of a story that was published this past summer, and I'm going to walk you through some of the results that we have in the story and how it's really convinced us about the importance of mechanics and stem cell function. So first of all, starting with what I said, OPCs, as they age, do not proliferate well, they do not differentiate well. Okay, so typically we can look at that these sorts of things in a simple way just by essentially looking at, for example, EDU pulses and counting the number of EDU positive cells. So EDU is essentially a cell cycle. It measures cell cycle. It measures whether or not the cells are in the cell cycle. So you can look for EDU and count the number of EDU positive cells over the total number of cells, in this case, cells in the oligodendrocyte lineage, and form this number called proliferation capacity. So what we can see here is that neonatal OPCs proliferate very well, aged OPCs proliferate very poorly, exactly as I told you before. And the same sorts of things with differentiation. We can look at differentiation markers and form this number called differentiation capacity. And again, neonatal OPCs have a high differentiation capacity. They differentiate well. Aged OPCs differentiate very poorly. So this is basically most of the data that I'm going to show you is really about proliferation capacity and differentiation capacity. This is kind of two sides of regenerative potential. So OPCs lose the capacity to proliferate and differentiate with age, and there's no known cocktail of growth factors that can reverse this. So the first thing that we asked is whether or not this aging phenotype can actually even be reversed by cell extrinsic factors. Is it just something cell intrinsic? And so in order to figure this out, we took, all the work that I'm going to tell you about is in rodent. In this case, it's in rat. So we could take neonatal OPCs and aged OPCs and put them in the neonatal rat. Now the expectation here is that if this loss of function is cell intrinsic, then the aged OPC should still not proliferate well, even in the neonatal rat. However, what we saw was that when we took neonatal OPCs and put them in the neonatal rat and aged OPCs and put them in the neonatal rat, the proliferation capacity was exactly the same. And in fact, the differentiation capacity was exactly the same. So there's nothing cell intrinsic about whatever's happening to these aged OPCs in terms of loss of function. You put them in the neonatal environment, and they become like neonatal OPCs, at least functionally. Now, of course, there's a lot of things that can be happening in the brain to do this, including growth factors, et cetera. The first thing that we asked was whether or not it's ECM. And so to figure this out, we developed these sorts of decellularized scaffolds in the brain so that we could get neonatal ECM and aged ECM and put neonatal OPCs on aged ECM and aged OPCs on neonatal ECM. And so here for your reference, here's neonatal OPCs, the proliferation capacity on glass. They proliferate quite well, as I said before. Aged OPCs on glass proliferate poorly. Aged OPCs on the aged ECM still proliferate poorly, as you would expect. But when we take the aged OPCs and put them on neonatal ECM, they proliferate very well. And in fact, they differentiate very well. And moreover, neonatal OPCs lost the capacity to proliferate and differentiate on aged ECM. So the microenvironment is regulating the phenotype here. Neonatal become like aged OPCs on an aged ECM, and aged OPCs become like neonatal OPCs on a neonatal ECM, at least functionally. So it was quite interesting thinking about this. Okay, what could possibly be going on here? when we look at the sort of aging extracellular matrix, what we see, chondroitin sulfate is one of the major ECM components, and we see that it looks quite a bit different going from neonatal to aged. In fact, it looks a bit more cross-linked in the aged. So what we thought was, well, what if we kind of enzymatically degrade the chondroitin sulfate and sort of break up the aged ECM? So this, we can use chondroitinase, which causes enzymatic degradation of chondroitin sulfate, inject it into the aged rat brain to break up the ECM a little bit and see if we can rejuvenate these the aged OPCs within the CNS. And what we found is that indeed we could. So in the rats that were treated with chondroitinase, the aged rats that were treated with chondroitinase, proliferation went up highly significantly, and differentiation went up in a highly significant way as well. So we have higher regenerative potential by enzymatically degrading the ECM. So kind of just breaking it up a little bit gives you a kind of regain of function for these OPCs in the aged animal. But I want to look just a bit more closely at this, and I sort of mentioned this already. The big difference between the neonatal rat brain matrix and the aged rat brain matrix is that the aged rat brain matrix, looks a lot more cross-linked. And so mechanically, you think if it's more cross-linked, then it's very likely that it is stiffer. And so we went and we performed atomic force microscopy in collaboration with Christian Franza, Christian Franza's lab here at Cambridge. And what we found is that throughout the lifespan of the rat, the brain stiffens considerably. So AFM measures stiffness. We could see that the aged rat brain was much stiffer than the neonatal rat brain, and the young adult is somewhere in between. And that's across all regions of the brain. So what I've told you so far is that aging in OPCs is not cell intrinsic. Aged ECM drives the loss of function, and aged ECM is stiffer. So that, I think, really begs the question, or really drives us towards the question, is it actually just stiffness? Maybe it has nothing to do with the ECM at all, except for whether or not it's stiff or not. And so this is what we can use these hydrogels for that I was telling you about earlier. We can control for stiffness. We can make hydrogels the same stiffness as the neonatal rat brain, and we can make hydrogels the same stiffness as the aged rat brain, and ask whether stiffness is driving loss of function. So we synthesized these hydrogels, and here's what we found. So again, here's the proliferation capacity and the control. Aged OPCs proliferate poorly. Neonatal OPCs proliferate pretty well. The aged OPCs on stiff hydrogels, which are stiffness matched to the aged ECM, still proliferate poorly, which isn't a big surprise. When we take the aged OPCs and culture them on the soft hydrogels, which are stiffness matched to the neonatal brain, all of a sudden they start proliferating very well. In fact, arguably better than the neonatal controls. And in fact, they start differentiating very well too. Again, just as well as neonatal OPCs. So just by putting them on these soft hydrogels, they start proliferating and differentiating just as well as neonatal OPCs. It's really quite a dramatic increase. It's essentially a binary effect in terms of stem cell function. And in fact, when we took neonatal OPCs and put them on the stiff hydrogels, they started proliferating and differentiating very poorly. So again, aged becomes like neonatal on soft, neonatal becomes like aged on stiff. This is driven purely by mechanics. And when we did RNA sequencing to see whether or not these cells were molecularly rejuvenated in addition to functionally rejuvenated, what we found was that, first of all, the OPCs on soft hydrogels clustered exactly with neonatal OPCs, and the OPCs on stiff hydrogels clustered exactly with aged OPCs. And when we do a sort of pathway enrichment analysis, what we found is that many of the pathways that are being enriched by putting these cells on the soft gels are the very same things that people are looking at in the context of aging. A regulation of autophagy, proteostasis, DNA repair, etc. These are the sorts of things that are being most enriched on soft gels. And this is quite incredible because there's really a kind of good match between what people have seen to be sort of mechanisms that are driving aging. So soft gels, whatever it is that they're doing it holistically. They're driving functional rejuvenation of the OPCs and molecular rejuvenation. So in some sense, ECM mechanics is overriding physiological age of these cells. So this is why we think stiffness could potentially be more important than perhaps people have even realized yet. That it seems to be, at least in this stem cell system, stiffness is very important for the aging phenotype. So we wanted to go for a little bit of molecular insight here. We now know that we can rejuvenate these cells. But there's obviously some things that you could do with hydrogels by perhaps putting them into patients and things like that. But we also want to think about, what can we learn? Is there something that we can target? Is there something sort of druggable maybe even that we could get out of this? Because as I said, there's an engineering aspect to this, but there's also an aspect of, what factors might be involved that perhaps we could start to use with patients. So in terms of ways to deal with this, there's two different ways that are clear. One is that if you want to try to kind of regain the function in these aged OPCs, you can kind of try to soften the ECM in vivo. Now, I already showed you that you can do this with chondroitinase, that it does actually bring some rejuvenation to these cells. But this is quite a strong thing to do. Perhaps the more clever way to do it would be to target the ability of the cells to sense their mechanical microenvironment by perhaps deleting some kind of protein. And it turns out that there's really quite a few candidates for that. There's things like focal adhesion kinase, which work with integrins. There's actin contractility in the cells, there's contractility of the cytoskeleton, and there's also mechanosensitive ion channels. And we investigated many of these factors and whether or not they could lead to a sort of rejuvenation effect. And in fact, what we did find is that if we reduce actin contractility using a small molecule called blebistatin, we found that we could take aged OPCs and give them much higher proliferation and much higher differentiation. So by essentially blocking actin contractility, we did get this kind of functional rejuvenation, or this regain of function in aged OPCs. We looked at other things like focal adhesion kinases and also lamin AC, which is another mechanosensitive protein in cells, and we didn't see much effect at all. So essentially some things worked and some things didn't. But there was one that we wanted to study a little bit more closely that looked a bit promising, and that is Piezol, which is a mechanosensitive ion channel. Now, Piezol it's a non-specific ion channel in the sense that it opens or closes in the membrane, and it lets ions in or out, depending on whether it's, and there's nothing specific about that. But it is very specific in the sense that it opens or closes with force. So essentially, when a cell is in a stiff environment, they tend to open, in a soft environment, they tend to stay closed. And abrogation of Piezol, we already know this, leads to, in many cell systems, decreased cell spreading and actin contractility, most likely due to reduced levels of intracellular calcium. And you can see here that when we look at calcium imaging on the aged OPCs on stiff hydrogels, and the aged OPCs on soft hydrogels, and the aged OPCs on stiff hydrogels, but with Piezol knockdown, what we see is that on stiff hydrogels, you see these calcium fluxes. On soft hydrogels, you don't see calcium fluxes. And when you put the OPCs on stiff hydrogels, but knock down Piezol, you don't see the fluxes. And that's all quantified here. So clearly, knocking down Piezol or putting cells on soft hydrogels causes less calcium influx. So that made us think, well, maybe we can sort of phenocopy the soft gel phenotype by abrogating or inhibiting Piezol function. And so we knocked down Piezol, and what we found in aged OPCs is that we got significantly higher proliferation and significantly higher differentiation, making this a pretty promising target because it's a single protein that we can perhaps inhibit and see whether or not we can get rejuvenation. And we do see this sort of increased regenerative capacity. And we wanted to see whether we could see this in vivo too, which is, of course, the ultimate goal here. So the idea is to specifically target Piezol with CRISPR-Cas9, for example, to sort of genetically engineer the cells not to express Piezol or to inhibit it. But unfortunately, we can't do that in aged OPCs because they don't cycle well, and it's very difficult to transfect them. So it's really very difficult to do this efficiently in aged OPCs and get the numbers that we need in order to inject aged OPCs that are genetically modified into the aged rat. However, we do have an interesting system, or we do have one promising possibility, though, because neonatal OPCs are relatively easily transfected, and they age immediately upon transplant into the aged rat CNS. So neonatal OPCs become like aged OPCs when you put them into the aged rat. I didn't show that data earlier, but it's similar to what I showed earlier with the stem cell transplants. So here, the idea then would be to sort of silence Piezol-1 in the neonatal OPCs, put them into the aged rat, and see if they can actually thrive within the aged microenvironment. And so to do this, we used a Cas9 knock-in for a shRNA for Piezol-1 in order to inhibit it. And what we found when we did that, so these are the neonatal OPCs that we transplanted into the aged rat. And what you see here, EDU is in red. This is the proliferation marker. You don't see any red because these cells are not proliferating. As I said earlier, when you put them in the aged microenvironment, they don't proliferate and they don't differentiate. So that's not a surprise. So this is the non-targeted ones. But in the ones where Piezol-1 is inhibited, we start to see lots of red. And in fact, neonatal OPCs with inhibited Piezol-1 are able to proliferate in the aged rat brain. So this is, I think, potentially very interesting from a sort of therapy standpoint, in the sense that here's a system, at least in this system, healthy progenitor cells do not proliferate in the aged microenvironment. Now, this is, of course, what's done in stem cell therapy, is you take stem cells and put them into, in many cases, an aged microenvironment. And, you hope that they thrive. But if it's the case that the microenvironment is driving aging, this could be a significant issue for stem cell therapy. So the idea that we have here and what we're pushing forward is to rejuvenate aged progenitors on hydrogels, like we've done, and then they're also back into the cell cycle, so you can genetically engineer them more easily to be insensitive to the aged microenvironment when transplanted. So, for example, and also if you need to mutate them in an additional way, it allows you to do that, and also allow them to be transplanted into the aged microenvironment and potentially thrive. So I think from a stem cell therapy standpoint, this could be very exciting. But this is in stem cell transplant. We also wanted to take this a step further and see if we could show true in vivo rejuvenation in these sort of resident OPCs by inhibiting Piezol-1. And so what we need is an OPC-specific Piezol-1 inhibition in the aged rat. So this is obviously not straightforward, and it's something that Mikey, the PhD student who was working on this project, worked very hard on. And basically, what we needed was a cell-specific in vivo CRISPR to inhibit Piezol-1 in the aged CNS. And to do this, he combined 3 synthetic biology techniques. One of them is using PHP-EB adeno-associated viruses that efficiently infect the CNS following 1 intravenous injection. Another technology recently published was this CRISPR strategy to knock in genomic constructs in adult post-mitonic cells in vivo. And last was to drive a guide RNA from a cell-specific RNA pull-2 promoter using ribozymes. In this case, we want to use as a promoter PDGF receptor alpha because it's specific to OPCs in the CNS. So in this way, we get delivery into the CNS. We can get these constructs into post-mitonic cells, like aged OPCs, and we can drive only from the promoter that's specific to OPCs, in this case, PDGF receptor alpha. So we combined all of these things, we validated them, showed high indel efficiency, and about a 75% knockdown of Piezol-1 in OPCs. And for all the validations and more information, you can see the paper. But importantly, when we did this, what we saw was that we were able to get, this is proliferation capacity here on the right. We got much higher proliferation when we knocked down Piezol-1 in the aged OPCs in the rat brain. We got much higher differentiation in the Piezol-1 inhibited aged OPCs. And perhaps most importantly, in a lesion model, we got significantly more remyelination in the animals with the Piezol-1 knockdown in OPCs. So what this says is that inhibiting Piezol-1 leads to significantly increased regenerative capacity in aged animals for OPCs. So, we can conclude from this that mechanical signaling is very important for OPC aging, and that we really need to be considering mechanics in the context of the functionality of these cells. So, stiffness drives aging in OPCs. A soft ECM alone kind of reprograms aged OPCs to be like neonatal OPCs. And finally, Piezol is maybe a very potentially interesting therapeutic target. Now, I wanted to finish with a couple of thoughts, because I think there's a couple of really interesting open questions here. First, why do these cells express Piezol to begin with? Now, I had assumed it was because, well, maybe if OPCs, Piezol may be bad for OPCs, especially in the context of aging, but maybe it's good for the differentiated lineage, like oligodendrocytes. But actually, it turns out that OPCs, as soon as they differentiate into oligodendrocytes, they silence Piezol. So obviously, it's important for the stem cells and not necessarily the differentiated lineage. But the next thing that we thought is, well, okay, it's bad for aging, but maybe it's really important for development. And so Mikey used a very similar strategy here to essentially inhibit Piezol in neonatal rats. Okay, so these are essentially just newborn rats that we inhibit Piezol to see what happens. And what we found, actually, is that proliferation in the neonatal rats, when we looked several weeks later, was much, much higher. And in fact, the cell numbers were much, much higher when we deleted Piezol, or when we inhibited Piezol. And so basically what this says is that OPCs significantly overgrow in this context. You essentially have too many. So it could be that Piezol or mechanical signaling in general is helping to regulate cell numbers and development. So even though it might be bad for aging in the sense that it leads to a loss of function in these cells, it actually does seem to be, maybe it could be quite important for development in terms of regulating cell numbers. Then another open question is why does the ECM get stiffer with age at all? It seems like a soft brain is better in this context, so why does it get stiffer? And so we were thinking a little bit about, immune capacity. And for this, we looked at primary astrocytes on soft and stiff extracellular matrix. And what we found, astrocytes are essentially inflammatory for inflammatory response in the CNS. And what we found is the astrocyte activity is highly suppressed on soft gels. It's much higher, the activity on stiff gels. So this suggests that even though a soft brain might be good for the progenitor cells and stem cells, it may not be so good for the cells that are giving you immune response and inflammatory response. So why do these cells express Piezol? It could be because it's needed for development. Why does the ECM get stiffer with age? Well, perhaps what's good for what's bad for progenitors and cells and stem cells might be good for inflammatory response and immune response. And just to kind of sum this up, in OPCs, we also know that the ECM production goes up and matrix metalloproteases goes down with age, and it suggests some kind of feed-forward loop. And these are the kinds of things that we might be able to reverse by putting these cells in the right mechanical microenvironment, or a better mechanical microenvironment. Just to kind of sort of, start to finish this up, or start to tie this up, I'm suggesting here that proper maintenance of the ECM microenvironment might be very, very central to aging. And it's going to be very interesting to see if this is true in other progenitor cell systems and stem cell systems. So back to mechanical signaling just for 1 second. We are thinking a lot about this, that maybe we need to really be controlling mechanics for stem cell progenitor cell function, for things like regulation of cell numbers, immune system activity or immune response, inflammatory response, also for aging. And the hope here is to get strategies to really kind of disrupt mechanical signaling in age-related pathologies, such as things like liver failure and multiple sclerosis, not only to give them a good environment to thrive in, to perhaps transplant into patients, or to just prepare them for transplant, but also to try to find sort of targets for getting them to work better once you do transplant. Okay, and I want to finish by thanking my lab, and in particular, I want to thank Mikey Siegel, who did almost all the work that I just told you about, and also Robin Franklin's lab, who is the major collaborator in this. So Robin and I co-supervised Mikey and worked very closely together to make this work happen, and Chabeza Agley for the hydrogels, and many others that were involved, and Elisa Molotova was also involved in some of the astrocyte work on Christian Franza and the atomic force microscopy. Thank you very much for listening, and I hope there's some good questions that I'm happy to answer. Thank you.
So thank you very much, Kevin, for the talk. I have to say, even myself, I think I was not aware that stiffness was that important. So now, yeah, so we're open to questions. So if there is any question, you can put it on the chat. Yeah, so it starts straight away. So if you want, Kevin, you can say the question and answer directly.
Okay, yeah, can you hear me okay, or?
Yeah, I can. So.
All right. Okay, so the question is, how long do the cells survive after transplantation in the rat? And have we taken the model out several months? The answer, it's a very good question. We actually don't know the answer to that. We certainly know that that they survive transplantation for a couple of weeks, but we haven't taken it out several months. We'd like to do some behavioral studies and things like this to try to see exactly, what happens in the long term. I think that's an incredibly important point, that we don't really know what these cells that where Piezol-1 is inhibited, we don't know what they'll do in the long term. So it's a very good question. Yeah, so the next question is Piezol-1 expressed in astrocytes? That's also a really good question, and I hate to say that I also don't know the answer to that. It's not expressed in that many cells in the brain. We actually do have, in the paper that I referenced, the paper that came out a few months ago that was referenced in the talk, we actually have done an analysis of Piezol-1 on many across the CNS. Astrocytes would certainly be included in there, and that the figure, which is in the extended data, would tell you the answer to that question. I believe that the astrocytes do express it, but as I said, that's in the paper. So I'll refer you to the extended data in there. And is it relevant to human stem cells? Well, we did at one point get some human samples, I think from what kind of patient? I can't remember, but it was just a one-off thing, so it has to be taken with a grain of salt. And what we did see is that we were able to get aged OPCs from a human patient proliferating again on the soft hydrogels, and it was quite a dramatic effect. But we really only did it once, and it's something that really needs a lot more, would need a lot more kind of work and thinking about like how to do this in the clinic. So there'd be a lot to do there to try to really address that question perfectly. But again, very good question. I suspect it is, honestly. I mean, we know that Piezol-1 is expressed in human OPCs, so I have no reason to believe that it wouldn't be beneficial there. So let's see. Are there antibodies to Piezol-1? Yes, there are. I think there's only one antibody that we actually got to work. It's not very easy to find antibodies for Piezol-1, but Piezol-1 is taking a bit of a star turn in the literature right now, so I think there's probably more and more good validated antibodies coming out on it. We did find one good antibody, and again, I'll refer you to the paper for that. It's in the materials and methods of the paper. Anybody who needs a copy of the paper, if they can't get access, please just email me, and I'll send you the relevant data. Happy to address any of these questions. Actually, for specific questions, if I'm not able to answer them here, just please email me, and I can give you more, better answers or send you the paper. So how is the StemBond gel different from the activated EasyCoat polyacrylamide hydrogels of Matrogen cell guidance systems? I don't actually know the answer to that because I don't know the recipe actually that's used in Matrogen. So I don't know exactly how the polyacrylamide is being functionalized. I think it's a really good question. I think we the hydrogels that we developed were bespoke because we wanted to know exactly what was going on there. We don't know exactly how it compares to all other hydrogels. We knew that there were some types of hydrogels, for example, functionalized with sulfosampa, that were not able to support the OPCs. But there's other types of hydrogels, and I think Matrogen is a different chemistry than all of that, which could well support it. The main message here in this talk that I would say is that, as long as you have, the Matrogen should work, StemBond should work, anything that you can do that your cells will actually attach to, and this is the big point. If your cells attach to it and they attach stably, then it could be really, really beneficial to try to think about looking at them on a stiff versus soft environment. So if you try Matrogen and your cells stick to it well, then I'm sure that it's going to be very beneficial. Especially for stem cells from soft tissue, like liver, brain, embryonic lineages. I think this is where you really see the night and day difference in your cell culture between soft and stiff substrates. Okay, where was I? Okay, what about piezo2? Is there any role, and what about stiffness in the periphery? So piezo2 is not expressed in OPCs, so it plays no role in an OPC function. Piezo2, interestingly, I mentioned in the talk that that when OPCs differentiate into oligodendrocytes, they shut down piezo1. Which was really a surprise to us, because as I said, we suspected that the reason they express Piezol is maybe because myelination is such a mechanical process. We expected to see that you would need some kind of mechanosensitive protein present there, and we thought maybe it's Piezol, but it's silenced as soon as they differentiate. However, Piezo2 goes up in oligodendrocytes as soon as Piezol goes down. So Piezo2 plays no role in OPCs, but it might play a role in the differentiated lineage. But we haven't checked into that. And I don't know the answer to the question about stiffness in the periphery. I apologize for that, but I think we only measured the central nervous system. Okay, so what is the thinking behind a stiffer ECM benefiting an immune response? Yeah, good question. I'd like to know the answer to that. The thinking in general is that I don't think that mechanical signaling is like, for example, EGF or FGF signaling or TGF beta, which are like really specific instructive signals. I think mechanical signaling is not instructive in that same way, where it tells the cell, okay, this is how you should function. What I think that mechanical signaling does is that it changes the way that cells respond to those signals, like TGF beta and FGF. I think if you put, for example, TGF beta cells in the presence of TGF beta, they'll respond to that very differently if they're on a stiff substrate versus a soft substrate. This is what I think this is what I believe very, very strongly. So I think that there's probably some, in terms of immune response, there's some signals that are obviously important to that, some that are well known. And I think that most likely the cells are just responding differently to those signals on stiff versus soft. And on stiff, perhaps in a way that makes them be able to migrate better and respond better, be more active. But I think it'll be interesting going forward, knowing what sorts of signals are present there and how the mechanics of the substrate might synergize with those signals. Okay, is there any phenotypic or behavioral differences in the Piezol knockdown OPCs and the wild type OPCs? Well, okay, so behavioral differences we don't know. As I said earlier, we haven't done any behavioral studies yet, but I'm not sure if that question is about cell behavior or organism behavior. But anyway, phenotypic differences, not really. They can still migrate, they still graft. No obvious differences. We haven't done any like sort of deeper analysis on that, but there's nothing obvious. We were surprised to see that they can still migrate. I showed that one image that after in the Piezol inhibited aged animals, where we did the sort of cell-specific in vivo CRISPR, we were really pleased to see that, in the lesioned area where we got rid of the myelination, that you actually saw the Piezol inhibited cells migrating into that lesion. So they seem to be, there doesn't seem to be any major problem with them, but, deeper analysis needs to be done there. Have you tried small molecule inhibitors of Piezol? There are no specific small molecule inhibitors of Piezol. People use this GSM TX4, which is some kind of tarantula toxin. Actually, it's not specific at all to Piezol-1. It just sort of changes the lipid composition or stiffens the membrane so that it makes it harder for Piezol-1 to open and close. But it changes a lot of other things too, because it's essentially changing the mechanics of the plasma membrane. So I wouldn't, there's nothing specific about it. So far as I know, I don't think there's any specific inhibitors of Piezol-1. Though with everything that's coming out about Piezol-1 right now, I'm quite sure that there are some people interested in drug targets that are working on this as we speak. Hopefully there'll be something too. But nobody, I'm not aware of anything. Okay, so let's see. Are there plans for work on human system, human OPCs? There are no imminent plans for it. I would need to have a collaborator for that. I'm not going to, you'd obviously need to, you'd need to work with somebody in the clinic for that. It's not something that, we're going to have any sort of access to. Obviously everything that we were doing in the paper was primary cells. So it's not that, obviously it's a whole different level to get, human OPCs in that respect. So I there's a lot of things that we get up to in my lab in terms of mechanical signaling, but in terms of the sorts of systems that we are looking at, we really rely on collaborators. So if anybody out there is interested, of the people on the webinar, if you work in human systems, let me know. Okay, and what about YAP-TAZ signaling in your research? We don't really look that much at YAP-TAZ. It doesn't seem to play a huge role in soft tissue, and we mostly work in soft tissue like brain and liver. So, we have certainly looked at YAP-TAZ. There's no real difference in the localization of YAP-TAZ in the OPCs that we have ever seen. We've never, seen anything to indicate that that there's any reason to look at it in our systems. Same thing with embryonic lineages, which we also look at in liver. My suspicion is that YAP-TAZ is more of a mechanical relay. It's not like an initial mechanosensor. And so like Piezol-1 is, or like maybe focal adhesions or some other things in the membrane. So I would think that, if you really want to go for a sort of targeted, efficient therapy, or any way to kind of block the mechanical signaling, it might be better to go for something that's more of an initial mechanosensor and not something downstream of that, like YAP. Okay, and is there anything to indicate that cancerous cells also respond to ECM stiffness? I don't know, actually. It depends on the cancer. There's this kind of tension, I guess, if you want, for maybe not the best word to use in this context, but anyway, there's this kind of tension that exists in cancer between the fact that you have in for example a tumor, you have to have some sort of self-renewal, where you have some mutated cell that takes over and starts self-renewing to make a tumor, but then you have to have the ability to get out of the tumor, some kind of for metastasis, cells have to escape the tumor. And I would imagine that these are very different, and this could be part of the problem. These are very different kind of mechanical things that are going on here. As I've shown here, a sort of soft environment seems to promote proliferation in this system, but that same soft environment is not going to be as good for migration. So there's a kind of push and pull there with cancer, which makes it a really interesting system. But I think there's probably some really interesting things to be studied there in terms of mechanics, but it's not going to be straightforward, just because you have to have proliferation and migration, so which are kind of two very different mechanical inputs into that.
Okay. Thank you, Kevin. I think it's been more than 10 minutes, actually. But it was really interesting questions, to be honest. So even me, I wanted to know the answer.
Yeah. Okay.
But yeah, thank you very much. So as everybody saw, it's pretty interesting. Stiffness definitely matters. So if anybody is interested, so of course I can put you in contact with Kevin directly. And if you would like to know a bit more about hydrogel, we'll be happy to answer a question about the one we have available now. So I hope everybody enjoyed the talk. We will probably do another webinar at the beginning of the year, so end of January, beginning of February. So I hope to see you all there. And thank you again, Kevin, for the time. It was really interesting.
Thank you everyone for coming along too.
You're welcome.